Hey guys, welcome back to Benzo. Today we're going to be talking about mitosis. It's the wonderful process in biology where you go from one cell to two cells. So, how does that happen? Is 1 equals 2? No. So, today we're only going to be covering mitosis. In the next video, we'll be covering meiosis. But basically, if we're thinking about how the cells arise, where did cells arise from? Well, we have the cell theory, which claims that all living cells, so all living cells that you have in your body, comes from pre-existing cells. And for the definition, uh, if you forgot the definition of cells, it's basically the basic units of life. So, if all living cells comes from cells, why do we need to do mitosis? Well, there's two main ideas. First, we need to make cells and cells and cells and cells for us to grow. So, growth is so important that although I I don't have the same size as I was in when I was 10, now that I'm 17. So all of the cells making it so I can grow, so I can have larger organs, so I can have more skin cells, it always goes through that. So mitosis is that cell division that is responsible for our growth. Next, we have the repairment of tissue. You know, when you get a cut, and then in 1 or 2 weeks, it's gone basically. Like the blood clot already formed, but another layer of skin cells were made, right? So all those skin cells, the tissue that you worn out, you know that, like, for example, we have over, if I don't forget, I think it's over 3 million red blood cells that die each day. So imagine remaking all of that, as you grow. So you need more of that. So cells making cells. Mitosis is cells making cells to perform functions of the organism. So here it's all about one organism, okay? Meiosis is different, right? Because you're making another organism. So, some key things that we need to know. We're going to go from one cell to two cells. Do you agree, if we're looking at the big picture, we can get this cell and make it so large that we can split into two cells that will be about the same size of that. So we would need twice the cytoplasm, which is the liquid that is responsible for maintaining everything organized. It's also, if I don't forget, it's also like rich in nutrients. So cytoplasm is, I think, one of the most important parts that we have in our cell. Uh, it is responsible for the shape of the cell. So we need to make 2 times the cytoplasm to get here one amount of cytoplasm and then another one amount of cytoplasm. We need double mitochondria, all those organelles. We need to make all those organelles. So we're making mitochondria, and mitochondria is going to represent, for example, mitochondria, the Golgi apparatus, which is when proteins are released to the outside in vesicles, the Golgi apparatus, the ribosomes. So we're, and etc., right? We're making all of that again. So everything can have, one amount of Golgi apparatus, one amount of Golgi apparatus, one amount of ribosomes, and one amount of ribosomes. So why does it have to be equal? Like, why does it have to be right equal? Because we're making sure that we have cells to perform the exact same functions as it was present on the first cell. So we want to make those exact same functions. And also, the most important is double DNA, the genetic material. So basically, imagine like this: we get a chromosome, which is held by this centromere, which is really important. It's a key term here, centromere. So center, it's where it holds these two sister chromatids, and we're gonna photocopy it. We're gonna make two identical copies of that. Okay, so basically, let's go here. So we have the human chromosome. There is 46 chromosomes, or two pairs two pairs of 23, which is equal to 46 chromosomes. We get one pair from mom. So mom, this is bad. So mom gives us 23, and dad also gives us 23. Okay, now the process here, we have this name of PMAT. Okay, so as you see here, this cell is way larger than the cell that we started here. If you remember how it was, like, it was the same size as here, right? It was the same size as this one. So we basically, we grow it a lot, then we start splitting genetic material, and then we make the two cells. So before we go to this division, which is called the PMAT, we need to look at the cell cycle, which happens in both mitosis and in meiosis. So we have the G1 phase. So before we have the G0. So it's when the cell is performing its function, but then something happens. You get a cut, or it sends a signal that you need to grow more, that you need to that the cells need to produce more so they can handle that, or making red blood cells, or making everything that you need to sustain. So the cell will go from G0 to then G1. The G1 is saying everything double. So the thing that I was talking about, everything you gotta double. Okay, now in the DNA synthesis, we get the chromosome. I'm sorry that it's a bit different from here, but it's the same. We get the chromosome here, and then we say, we gotta make sure that we get the same amount of chromosomes for each cell, so we gotta double. Okay, so I like to remember the S phase. So we have the G1, we double the cytoplasm, double the organelles, double the microtubules, which is really important because they make what we call the spindle fibers. And this spindle fibers is what attaches to the centromere and pulls it apart, pulls the chromosome apart. And then finally, after, so we have double cell. Okay, we have everything that a cell has but double, and we have this double genetic material. Then in the last phase is the G2, which checks for damage in the chromosome, and if the damage is so bad that it cannot be that the damage cannot be reversed, this cell triggers apoptosis, which is when the cell dies. So let's look at this diagram so we can understand the cell cycle. Okay, so we have the GO phase, which is the resting phase. Then we go to the G1, where the cell grows. Then we have the CS phase, when the DNA replicates, and DNA replication is an important concept. So remember, it's about breaking. So you have here, let's say you have your DNA here in this spiral structure. I'm sorry that is bad, but you're breaking with the helicase, the hydrogen bonds, then you have them in the straight lines like this. Then you have, let's say, A binding with T and G binding with C. And then we have that DNA polymerase that makes the that we have the helicase, which breaks it down into replication forks, which is something that we already have a video. So if you want to remember, we have some also IB questions about it, so please go watch it. And in the G2, the cell prepares for mitotic division, which is when it divides. Okay, so now we have the prophase, the metaphase, the anaphase, the telophase. Probably one of the most exciting processes in all of biology, because otherwise we would still be one cell, and bro, we're 37 trillion cells. So imagine that cell starting dividing. So that's really interesting. So if you don't have enough time to watch this video, I just want to show this, that I think it's very interesting. So we got the prophase, the metaphase, the anaphase, and the telophase. Ways that you can remember: the P will be for pack, the M will be for middle, the A will be for apart, and the T will be for two nuclei. So you're packing the genetic material into chromosomes, okay? Instead of being chromatids, which is like this lines, we're packing it so it can protect the genetic material, protects it. In the metaphase, the genetic material aligns in the middle, and then we're gonna get the spindle fibers, which those spindle fibers are gonna pull them apart in anaphase, and in telophase we're gonna have two nuclei reforming, and we're gonna lead to cytokinesis, which is splitting the cell membrane into two identical cells. Okay, so let's go here. So prophase, we already have that DNA replicated, right? We already have. So before it was like this, and now there's this. We have the microtubules here, which was only two. Now there's four. We got some organelles here, right? We have some mitochondria. So we have the first step is that the nuclear membrane is going to disintegrate, which is where it holds the genetic material so it can move outside, right? Imagine we're getting this to go to the top so we can make a cell here, and we're getting this to go here so we can make a cell here, right? So the chromatin, which is the DNA like this, is condensed into chromosomes, which again, it protects the genetic material. Then we have the microtubules, which is going to organize the centrioles, which was that organelle that will help pull it apart, pull this chromosome apart into sister chromatids. So we have centrioles that stores the spindle fibers, and they're duplicated during interphase. Okay, so it's just a recap that the prophase is where you're going to pack. You're gonna pack everything in the middle, and this membrane here is going to disintegrate so the genetic material can move to each side, so you can move to each side. Now, in the metaphase, that it's all about lining up in the middle. So the chromosomes line up at the equator line, That's something that's the terminology. So you can, if you look at the map, you see the equator line. So just remember that. And the microtubules, look, they release some spindle fibers. And these spindle fibers, they attach to the centromere of the chromosome, where it's going to equally split it. So if you see the red here, this part is going to go here and this part is going to go here. So we're attaching it, and the anaphase is where you perform the stretch, the shortening, right? You're shortening the the spindle fibers. So the spindle fibers separate the homologous chromosomes into sister chromatids. So now, instead of them looking like a chromosome, they're looking like a chromatid. They are identical, right? They are identical because they were photocopied during they were photocopied in the S phase, right? Where there was the DNA synthesis, a better image here, right? That were photocopied in the S phase. Now that we separated and we have the genetic material on one side here and one side here, what happens next? Well, a nuclear membrane starts forming around the genetic material, right? The chromosomes. Oh, so imagine we had that chromosomes, right? We had this part here, and now it's going to decondense into chromatins, which is how it functions, right? Because we need those lines so we can read and make proteins. So the nuclear membrane starts forming around it. The chromosomes decondense into chromatin, and you can see how it's taking up the form of two cells. And the only thing that we need now is to make sure that this here is almost like a cut, right? Cytokinesis, which is the next part, which is where we're going to cut. So cytokinesis is the end, where in animals and in plants it's a different process. So in animals we call this the clavage floral, which is made up of this contractile ring that will pinch the cell membrane together. So imagine you have a bread, okay? And then you press it with all your strength in the middle, and you see that will take up the format of like almost like two breads because it's split in half. Now, when you cut the bread, you're gonna almost like have two breads, right? So this is what's happening. We have this contractile ring that pinches the cell membrane together until you have two cells. And these two cells are genetically identical daughter cells. Whereas in plants, you have some vesicles. So in plants, you have some vesicles that will release the cell plate in the middle. So we'll form a cell plate, and it's also going to form two plant cells. So for mitosis, this is the SL part of the biology course, where in prophase you want to pack everything in the middle. In metaphase you want to line in the equator line, and then in the anaphase you want to separate them, aiming to already construct a nuclear membrane, which is exactly on the telophase. And then we have cytokinesis, which splits the two daughter cells. And remember, in animals, we have the cleavage furrow made of this contractile ring, which is also made of actin and myosin. Actin and myosin, which is going to cut, right? We got the bread, and then you make two breads. So that's all for this lesson. I hope you guys enjoyed, and comment if you guys have any doubt, and I'll post some IB questions in the meiosis video to see if you guys understood mitosis. So see you guys. I hope you guys enjoyed, and Benzo out.